Manisha, are you having any trouble?
Actually, I'm trying to share my poster. Please wait a second.
Okay.
Hello. Isha, can you see my poster?
Yes, your poster is visible.
Okay, so now can I start?
Yes.
Okay. Hello, good afternoon, everyone. So, our topic of presentation is removal of heavy metals and volatile organic compounds by biofilter and bioabsorbent. As we all are aware of conventional methods of heavy metal treatment from wastewater, such as reverse osmosis, chemical precipitation, flocculation, and ion exchange. But our concern in this poster is to introduce an alternative to these methods. As you may be aware that these conventional methods are not efficient as well as are not economical, as these methods generate a lot of secondary waste which leads to pollution. So the methods such as biofiltration and bioabsorption, which involve employment of microorganisms both in living or dead form, such as bacteria, fungi, microalgae, have been found to serve the alternative purpose. If we talk about the recent research studies, then the recent research is more focused towards the bioremediation of heavy metal and volatile organic compound. So we have tried to gather the reports on the studies which are carried on the employment of such microorganisms such as fungi, bacteria, algae, all together on a single paper. From last decade, microorganisms have been used in the fermentation industry for the production of food products such as milk, cheese, meat fermentation, yogurt, and antibiotic production, alcohol production, beverages production, enzyme production, and hormone production. And there are numerous many more products which are being produced by the employment of microorganisms. But in recent years, microorganisms have been reported to show absorption accumulation of heavy metals internally and externally due to some biochemical processes which are occurring in them. The microorganisms are now being employed to filter the wastewater due to the efficient recovery of metals from them. For example, if we talk about the fungi, let's say for example, the Aspergillus species in fungi have shown remarkable absorption efficiency for heavy metals such as nickel, mercury, and lead. Next in this section, if we talk about the bacterial strains, then the bacteria which are found in metal contaminated area have shown metal tolerance and also the metal absorption and accumulation efficiency in them. For example, Bacillus species is the most studied species for the absorption and accumulation of heavy metals in them, both externally and internally. For example, zinc, cadmium, cobalt, and mercury. These heavy metals have been shown to be accumulated in Bacillus species. Next, if we talk about the microalgae further in this section, both green algae and the brown algae have shown capability for the absorption of heavy metals in them. For example, Chlamydomonas, chlorella, etc. Next, if we talk about the role of microorganism in the elimination or we can say bioremediation of volatile organic compound, then there are numerous examples of bacterial species such as Pseudomonas, Bacillus, Staphylococcus, and Rhodococcus. These are the most studied and employed microorganism species till now. Since these species possess a number of oxidoreductive enzymes in them, that help them in the degradation of volatile organic compounds. So these species can be employed for the bioremediation, or we can say elimination or degradation of volatile organic compounds which are released from the sewage treatment plant or wastewater treatment plants. Next, if we talk about the role of the plants in the heavy metal elimination and volatile organic compound remediation, then there are a numerous species of plants which accumulate heavy metals in them. And about the plant, my partner Vikas will tell you the further details. So over to you, Vikas.
Apart from the microorganisms, some plant species have been reported to show accumulations of heavy metals in them. For example, Phylacus emblica in many studies have been shown extraordinary absorption efficiency of cobalt, nickel, manganese, lead, etc. Along with this, plants have been reported in elimination of volatile organic compounds present and released from the wastewater treatment plant. For instance, various ornamental species such as Chlorophytocorosum are useful in elimination of volatile organic compounds. Now, I would like to thank you all for listening us so patiently.
Thank you, Vikas. Now I would like to request the judges to ask any questions if they have any.
Yes, Manisha, are you hearing me?
Yes, sir.
Yeah, yeah, you have presented very well. Really, it's an informative session. But, since I'm not a
Thank you
biologist,
so much, sir.
but still I have a query in my mind, whether this microorganism is our friend or you can say it's our enemy.
Sir, this is the if we talk about the sir, we can say that it is our friend and enemy as well, because there are a number of microorganisms which are pathogenic to human beings and animals and plants also. But if we talk about some species which we can employ for the betterment or for the production of compounds or metabolites which are very useful for the human beings. For example, sir, if we talk about the curd formation, lactobacillus is the bacteria that is responsible for the curd fermentation. So, sir, there are numerous bacteria which can serve as the purpose of our friend, as well as there are pathogenic bacteria. So we can say microorganism can be our friend, but we need to identify. And if we want to make any pathogenic bacteria our friend, then we have to manipulate its DNA. By genetic engineering, we can also make a pathogenic bacteria our friend or any other microorganism our friend. We
Okay,
can employ that.
right.
Yes, sir.
Yes, because are you hearing me?
Yes, sir.
Yeah, because there is one, very simple query that we have discussed about the plants. So
Okay.
approximately how many species of plant might be there? Approximation, some idea?
Sir, I've gone through all the research papers. Shows that the Phylacus emblica plant known as Amla and the basal plant that's a neem. These are the plants which are helpful in restoring the power of and they are also helpful in elimination of heavy metal compound and volatile compounds.
Okay, that's fine. But in general, so many species of plant are there. There are n number of species might be there. So any idea if you collect all these plants in totality, then approximate how many numbers you can give? Any idea?
So know about the numbers about.
Okay, yeah, that's from my side. Yes,
Sir, excuse me, sir.
yeah,
Sir, I would like to answer the question on the behalf of Vikas.
yeah.
Sir, as we have read various research papers, so in my opinion, there are more than 25 plant species available
Okay,
about which we have read.
right, right. Okay, that's from my side.
Thank you, sir.
Thank you so much, sir.
Now, Now, I would like to request Akansha Kar Pathak and Simran Singh to please start with their presentation. And meanwhile, Pramender Shukla and Shalini Singh, please be ready with your presentations. Akansha Kar Pathak, please unmute yourself.
Yes, ma'am. Wait a second. We are sharing our screen.
Okay, thank you.
Good afternoon, everyone. Firstly, I would like to thank all the eminent speakers, teachers, and students for sharing their knowledge with us. I am Akanksha Karpathak.
And I am Simran Singh.
We would like to present our poster whose title is Review on Removal of Microplastics from Water Sources by Using Brown Algae.
So start with introduction. Basically, microplastic are fragment of any type of plastic less than 5 millimeter in length, as mentioned in our poster. They cause pollution by entering natural ecosystem from a variety of sources, including cosmetic, food packaging, clothing, and industrial processes. As we know, plastic degrades slowly, often over 100 to 2000 years. Microplastic have a high probability of agitation, incorporation into an accumulation in bodies and tissues of microorganisms. Microplastics are nowadays a great concern for an aquatic life. After they are detected in aquatic environment, they have become one of the emerging contaminant of growing concern. The removal of this contaminant from the water sources is urgently needed, but because of their size and properties, it is difficult to do so. Our study shows an efficient method of removing microplastic by using brown algae such as Fucus vesiculosus. Objective: basically, the main objective of our work is to find a good method for removing microplastic from the water sources. To identify various sources of microplastic pollution. Now, I would like to request Akansha to continue with the presentation. Thank you.
Thank you, Simran. Now, comprising the class of Phaeophyceae, as we all know what a brown algae is. Brown algae are a large group of multicellular algae, including many seaweeds. They also play an important role both as food and as potent habitat, as they are mainly found in marine environment, of course, because of that. Now, the type of algae we have studied in our research is Fucus vesiculosus. Now, Fucus vesiculosus, which is also known by some common names such as bladderwrack, red fucus, rockwack, etc. Now, the structure of Fucus vesiculosus consists of holdfast, which is attached to a substratum, and the rest of its parts are stipe, midrib, frond, receptacle, etc. It has ostiole and air bladder on its leaf, which helps in spore discharge and buoyancy of particularly. Now, in our study on getting a small cut on the leaf of vesiculosis, Fucus vesiculosus secretes a gelatinous substance. This gelatinous substance has alginate compound in it, and this alginate compound has anionic properties, because of which it and absorbs positively charged microplastics such as polystyrene, which is positively charged microplastic. Now, after the review paper we have studied, in result it was observed that approximately 94.5% of microplastics were removed by brown algae, and mainly positively charged microplastics were removed, as positively charged microplastics were attracted by negatively charged alginate compounds. So because of which the microplastics were removed from the area. Now I will conclude with my presentation. In our study, we have found that approximately 94.5% microplastics were removed with the help of algae, and out of various microplastics, the percentage of polystyrene microplastic removal was highest. But still, further studies are still needed for the complete removal of microplastics from the water sources. Thank you, everyone.
Yeah.
Now, I would like to request a judge to please ask the question if they have any.
Yeah, Akansha and Simran, it was really a nice presentation, and you have picked a very, you know, booming topic, that is the filtration and the water treatment of water. So one query is in my mind that we have used the term microplastic for some water filtration and all. So can any might be a difference that you want to use these microplastics from the ocean water and the river water?
So, I don't think we can use this microplastic. Of course, exceptions are always there. Maybe in future we can use it, but at right time it is causing us harm. So I think we don't. We just it is more importantly to remove it from the water, but. सर वी कैन यूज इट फॉर डेकोरेशन पर्पस आई गेस और एनी अदर पर्पस अगर जो रिसर्च फर्दर ऑन होती है तो फिर क्या पता हम लोग फ्यूचर में हम लोग उसको कर सकें यूज
Yes. Yes. Simran, have you heard about the term electrocoagulation, EC?
Yes, yes, sir.
The wastewater, what is it? Hello. Yeah, are you
सर
hearing?
इलेक्ट्रोकोगुलेशन जो है वो ब्रॉड स्पेक्ट्रम ट्रीटमेंट टेक्नोलॉजी है
Right.
जिसका हम यूज करते हैं सर रिमूव करने के लिए टीएसएस को, हैवी मेटल्स को और इमल्सिफाइड ऑयल को, बैक्टीरिया को एंड कंटामिनेट मतलब वाटर को, मतलब वाटर से कंटामिनेट को रिमूव करने के लिए.
That's from my side. Thank you. Thank you all.
थैंक यू सर
Now, I would like to request Pramendra Shukla and Shalini Singh to please start their presentation, and meanwhile, Megha Singh, please be ready with your presentation.
Thank you, ma'am. Let me just share my screen. And can you see my screen? Hello?
No, yes, it is visible.
Okay, thank you. Good afternoon, all. Me, Pramen Shukla, with Shalini Singh and Megha Singh. We have researched on the topic of is Omicron variant BA.2 is more serious than other variants. Now, Shalini will give you an overview of SARS-CoV-2. Shalini, please.
Thank you, Pramein. As of now, there are 42 million confirmed cases and 5 million deaths approximately pronounced due to SARS-CoV-2 globally. A 55-year vintage character from Hubei province in China may additionally were the primary man or woman to whom contracted COVID-19. The disorders because of new coronavirus spreading across the globe, and the cases dates back to November 17, 2019, in keeping with South China Morning Post. And now the variants of COVID-19 would be shared by Megha Singh.
Thank you, Shalini. Now let's talk about its variant. When virus begin to copy the mutated version of the virus, they create new variants. Variants are different than mutation. A variant is a copy of the original virus that has one or more unique mutation. First variation reported was 20I alpha version 1 and 20H beta version 2. 20J gamma version 3 was denoted by the Omicron as shown in figure 2. Now let's come to our topic. As per reports, first case of Omicron was reported in South Africa on 24 November 2021. Omicron is was mutated SARS-CoV-2 variation with the almost 50 transformation MS in its genome, and it is specific interest and varies since 26 to 30 changes in viral spike protein area, and out of these, 15 in the receptor binding domain. Cryo-EM primary investigation of the Omicron spike protein ectodomain shows the general association of the trimer is like the notice for the hereditary strain and every prior variation. Presently, another sub-variation of Omicron is overwhelming the cases in South Africa and Denmark. Omicron variation BA2 started from Philippines, South Africa announcing 100% of its everyday instances of variation 2. As indicated, the World Health Organization report variation BA2 is more contagious than variation BA1. Now come to Pramin, tell me the
Thank you, Megha. Now I would like to explain you about the mutation of Omicron variant BA.1 and BA.2 and difference between both of them. Omicron variation BA.2 and variation BA.1 share 38 nucleotide and amino acid mutation. However, Omicron variation BA.2 has an extra 27, while BA.1 has 20 extra. Looking similarly as Spike, both offer 21 amino acid mutation, with BA.1 conveying 12 extra interesting amino acid mutation and BA.2 conveying 6 extra. The 6 extra spike mutation in Omicron variation BA.2 are ST19I, SV213G, SS371F, ST376A, SD405N, SR408S. On the top of this, there may be a 9 nucleotide deletion from position 21633 to 21641, which ends up in the deletion and mutation SL24, SP25, SP26, and SA27S. As with other deletion mutation, this is every so often rather said as SL24S observed via 3 deletions. The ensuing protein is same. Some of the major differences between both the Omicron subvariant BA.1 has 39 mutation, BA.1.1 has 40 mutations, BA.2 has 31 mutation, and BA.3 has 34 mutation. With 21 shared mutation among all, Omicron sublineages BA.2 and BA.3 have a better transmission potential than BA.1, and BA.1.1 mutated resides in BA.1.1 K478, BA.2 R400, R90, R95, 495, and BA.3 R397 and H499 formation of latest salt bridges and hydrogen bonds. According to the Outbreak Information website, the cumulative prevalence for BA.1 is 8%, detected in at least 135 countries. BA.1.1 has 5%, detected in as a minimum 69 countries. BA.2 is 1%, and it is detected in least 69 international locations. BA.3 is much less than 0.5%, detected in as a minimum 16 international locations as of 10th February 2022. BA.1 rapidly became dominant and has tested considerable breakout from neutralizing antibodies, NABs, precipitated with the aid of vaccination. For BA.2 has these days extended in frequency in multiple areas of the arena, suggesting that BA.2 has a selective advantage over BA.1. Among the 34 new amino acid modification inside the BA.1 spike, 15 are within the receptor binding domain RBD, representing 7% of adjustment. BA.2 has BA.2 and BA.3 proportion 14 of the 15 RBD adjustment of BA.1. Thank you all. Now I would like to call Shalini for telling you about the future aspect of COVID-19 Omicron variant BA.2.
Excuse me, Pramendra, your time is almost up. Will you please wind up fast?
Yes, ma'am. Just give me 1 minute.
Okay, we will wind it up right away. As per all the examinations done, they show that there is no seriousness changes in the variation of BA.1 and .2. In the view of greater contagious BA.2 can be distinct advantage since it is hard to identify and can attack invulnerable framework without any problem. despite the fact that Omicron variation BA.1 and BA.2 is seen while quality sequencing is done in India. There were no reports now for Omicron BA.3 variation. We can't say that this is the last variation of SARS-CoV-2 since it is transforming its protein structure. Thank you, sir. And if you have any queries.
Yeah, a very nice presentation, and past 2 years we all are directly affected. The whole world is in the uh, COVID-19 has affected. So one query, one thing in my mind, that is there any prediction? Is there any mathematical model? Is there any kind of analysis by which we can say that what might be the stopping time, or what might be the ending of this particular virus and its variants? Is there any study about that?
Sir, as per the reports, they said that this will become an epidemic, not a pandemic anymore, and their mutation will be continue in future.
Right, right.
The only protocol that we should follow is like full vaccination till all age groups and all. Then this is only the way that we can convert it in pandemic into an epidemic.
Okay, fine, fine. And one more concern is that since we are talking about different viruses, different variants, so have you heard about the term gene variant?
Yes, sir.
So, what kinds of gene variants might be possible? Any idea?
Sorry, sir, we have
Okay,
no idea for this.
no problem, no problem, no problem. And the last thing is that since Omicron, Delta, and all these variants comes to us, so can you suggest us that besides having the vaccination, say booster dose also, say, is there any chances to get be affected by anyone, or severity of affection? Any idea?
Uh, Sir, there can be severity because, as you can see, Omicron is a much more mutated variant of COVID-19, and in future there will be more variants which can cause severity, and they can also be contagious.
Okay, okay. Thanks. This from my side. Thank you all. Thanks for nice presentation.
Thank you, sir.
Thank you for the questions, sir. Now I would like to request Kshitij Sharma to please start with their presentation and Pallavi Srivastav to please be ready with your presentation.
Thanks a lot, ma'am. Ma'am, just give me 1 second. I want to share my screen.
Okay.
Ma'am.
Ma'am, is my screen visible?
Yes, your screen is visible.
Thanks a lot, ma'am. My topic is application of CRISPR-Cas9 technology for editing of apollon to treat cancer. So we know that there is no effective way to treat aggressive cancers like brain and ovarian cancers. Chemotherapy resistance can be caused by a number of factors. Some of the cells that aren't killed by chemotherapy mutate and become medication resistant. There may be more resistant cells than cells that are vulnerable to the chemotherapy once they grow. Inhibitor of apoptosis, shortly known as IAP, is a family of proteins that has 1 to 3 bacilovirus IAP repeat domains and inhibits apoptosis by direct binding and inhibiting caspase. We found a human IAP family gene which we named Apollon, encoding a huge protein approximately of the size 530 kilodalton that contains a single BIR domain and a ubiquitin conjugating enzyme domain. Apollon protein was expressed in 4 of 6 brain cancers and 1 of 5 brain cancers in 38 human cancer cell lines that we examined. Among the brain cancer cell lines, SNB78 expressed a high level of Apollon, and this cell line shows resistance against various anti-cancer drugs. Treating SNB78 cells with antisense oligonucleotide against Apollon reduced the expression of Apollon and significantly sensitized the cells to apoptosis induced by cisplatin and camptothecin. Results conclude that Apollon protects SNB78 from undergoing apoptosis and at least in part plays a role in tumorigenesis and drug resistance of this cell line. Apollon level is important for apoptosis regulation. STR-A2 catalytically cleaves apollon with its serine protease activity. Apollon and STR-A2 mutually downregulate each other. In apollon-deficient cells, however, expression of catalytically inactive STR-A2 mutant with IAP binding motif also induced apoptosis. These results indicate that STR-A2 induces apoptosis in two different mechanisms, one with serine protease domain and the other with IAP binding motif in Apollo-deficient cells. The CRISPR-Cas9 is a versatile gene editing tool and a potent weapon in the arsenal of tumor treatment. So we needed a treatment for this because there is no other effective treatment in to treat the aggressive cancers. So CRISPR-Cas9 uses specific proteins to convalesce mutations at genetic level. In CRISPR-Cas9 system, RNA-guided Cas9 endonuclease harness gene mutation, DNA deletion or insertion, transcriptional activation or repression, multiplex targeting only by manipulating 20 nucleotide components of RNA. The researchers developed a novel lipid nanoparticle based delivery system that cancers that targets cancers and carries a messenger RNA mRNA encoding the CRISPR enzyme Cas9. Once inside cells, the Cas9 enzyme is assembled by host cell machinery and act as molecular scissors, introducing cuts to the cell's DNA strand that neutralizes the cancer and prevent it from replicating. They called the system CRISPR-LNPs. To examine if the novel therapy could be effective against aggressive cancers, the scientists chose to test CRISPR-LNPs in the mouse models of the two deadliest human cancers, glioblastoma and metastatic ovarian cancer. In glioblastoma, their average life expectancy doubled, improving their overall survival rate by roughly 30%. And in the treatment with cancer, CRISPR-LNPs in a metastatic ovarian cancer mice model increased their overall survival rate by 80%. So all this research and the studies only conclude that if anyhow we became successful in editing the gene coding for Apollon using CRISPR, then we would be able to treat different types of cancers and ovarian cancers which are very aggressive in nature. Thanks a lot, sir.
Yeah. Hello, Kshitiz. Really, it's a nice presentation, right?
Yes, sir.
See, cancer is a very deadly disease, and the number of patients are increasing day by day, and even uh, right now there is no age group at all. So it's a very challenging issue for all of humankind. So one point comes in my mind that in cancer, chemotherapy is a very painful process, right? All of you are aware with that. So is chemotherapy reduces the human life?
Sir, chemotherapy sometimes can reduce the human race because it has side effects, but in the treatment of cancer, it is also very effective. But but now the cancers are getting mutated, so chemotherapy is also not getting properly effective. So, sir, because of this, chemotherapy can take your life, sir. It can be dangerous to the life.
Okay. One thing more that can you name some of the types of chemotherapies, drugs,
Sorry, sir. Can you repeat?
the types of chemotherapy drugs that might be given to the cancer patients?
Sir, types of chemotherapy, doc.
Yeah.
Sir, I don't know much about it, but I have heard two to three types of chemotherapies, like alkylating agents, one and like anti-metabolites, I guess, or hormonal agents like that. And there was one more, anti-tumor antibiotics. These were the chemotherapy drugs, I guess.
Yeah, yeah, yeah. That's from my side. Thanks. Thank you.
Thanks a lot, sir.
Thank you for the question, sir. Now, I would like to request Pallavi Srivastav to please start with her presentation. And meanwhile, Pratibha Tiwari, please be ready with your presentation. Pallavi?
Is my screen visible?
No. Pallavi, please share your screen.
Already shared, solution.
No, it is not visible and your voice is also not clear enough.
Uh, Let me share it again.
Okay.
Is it visible now?
No, it is not visible.
I don't know what is the problem because I am sharing it continuously.
What is the problem? Yes, I think it is getting shared. Yeah, the PPT is visible.
Thank you.
Please start.
Good afternoon, everyone. I am Pallavi Srivastav, and I have got this golden opportunity to present my review work on the topic mucormycosis among COVID-19 patients in India. We know that we have lost thousands of lives due to COVID-19. We and we were only dealing with this global pandemic when one more deadly infections arose, making the situation worse. And name of this infection is mucormycosis, or generally we call this as black fungus. Mucormycosis is a rare but deadly infection caused by the fungi of order Mucorales. This function generally is spread through the inhalation of spores present in the air. Its main targets are the body parts such as brain and sinuses, stomachs and intestine, lungs and skin. Um, Symptoms associated with the mucormycosis are fever, shortness of breath, coughing, blockage of nasal passages, disturbed mental health, and irritation around nose and eyes. And if the infection keeps on advancing, it causes severe headache and even permanent blindness. So, the people who are more suspected to mucormycosis infection were reported to have weakened immune system or compromised immune system. It is often reported in patients with uncontrolled and unmanageable diabetes, and it's also reported in patients who are under treatment with high steroids for COVID-19. When the second wave of COVID-19 hit India, there was a drastic increase in the cases of mucormycosis infections. As of reported on 15 July 2021, India has observed over 45,000 infected cases and about 4,200 death cases of these fungal infections in patients which are mainly infected from COVID-19. Many of the patients that were infected from diabetes or were consuming very high powered steroids for the treatment of COVID-19. Overuse of and consumption of steroids escalates the blood sugar level, leading to compromised immune system, resulting in the widespread of this fungal infections. Also, the consumption of high amount of steroids weakened the immune system, making them more vulnerable or prone to COVID-19. This is not just one condition which is responsible for the widespread of this infection because it is linked with various medical conditions that promote its spread, such as stem cells or organ transplantation, neutropenia, which is the abnormally lower count of WBC, which is caused by damaging which damage the bone cells, bone marrow, and infections, and certain medications, trauma and burns, renal insufficiency or kidney failure, in which kidney loses the ability to remove waste and balance fluids. Broad spectrum antibiotics such as deoxycycline, ampicillin, and azithromycin. And finally, malnutrition, which is one of the major problem in India. The objective of this review is to highlight the reasons of the widespread of mucormycosis or black fungus amid of the global pandemic COVID-19, creating a pandemic within the pandemic situation, and also the use of various multidisciplinary methods for the remedy and prevention from mucormycosis. From this review, we draw the following results that mucormycosis targets the individuals who have uncontrolled and unmanageable diabetes, immune
no compromised conditions due to surgeries, and finally, the people who are consuming heavy steroids. People with weak immune systems are more prone to mucormycosis infections. Last, but the key point, it is more commonly reported in patients who are already infected with COVID-19 as the heavy steroids used in the treatment of COVID-19 promotes the growth of this fungal infection. Concluding my review work, I would like to say that mucormycosis has created a state of fear and panic in the whole uh, world along with COVID-19. But by taking proper measures and early diagnosis, we can eradicate these fungal infections. Discontinuations are the measures we can take to reduce these infections. By discontinuing the immunomodulating drugs and steroids, proper treatment and of diabetic ketoacidosis and radio imaging, antifungal prophylaxis, which is used with high rates of invasive fungal infections. And last but not the least, be determined to live. If you are determined to live, you can beat every difficult situation. So don't panic, be positive, and stay strong. Thank you.
Yes, Pallavi, are you hearing me?
Yes, sir.
Yeah, really, it's a nice presentation. And this black fungus, even the persons who was recovered from COVID second wave, this black fungus was a great, uh, thing that feels that recovery also. So, one my question is that in your conclusion, you have shown the radio imaging thing by which we can avoid this. So, how can we do that with that radio imaging?
Yes, sir. Because we have seen in the symptoms that it causes coughing and blockage of nasal blockage. So it is obvious that it is affecting the lungs, and we can X-ray the lungs to find out where is the infection is accumulating, and we can clear the infections.
Okay. So, this this there are different kinds of fungus, or you can say fungal infections are there. So,
Yes,
what might be the basic treatment of a fungus?
the
Uh,
most basic
yes.
treatment is to maintain hygiene. It is the most basic treatment. Maintain hygiene around yourself and don't come in contact or use the contaminated stuffs, and try to avoid the contaminated things that is used by the infected person.
Can you give some examples of fungus?
The ringworm is also caused by the fungus.
Okay. Thank you, Pallavi. That's from my side.
Thank you, sir.
Thank you for the questions, sir. Now I would like to request Pratibha Tiwari to please start with their presentation and Soumya Srivastava to please be ready with her presentation.
Is it visible? Hello?
No, it is not visible. Yes, it is
Is
visible.
it visible?
Yes, it is visible.
Okay, okay. Good afternoon, everyone. This is Pratibha Tiwari, final year MTech student of ILM Greater Noida. My topic for presentation is direct implementation of cellulase enzyme in sorghum feedstock for biofuel application. I'm starting with the cellulase. What do you mean by that? Cellulase is a biological catalyst used for the saccharification purposes in bioethanol production from lignocellulosic feedstock. Basically, it is a plant-based feedstock in which different carbohydrates are present, that is cellulose, hemicellulose, and lignin. It plays a very important role for the production by degrading the components which are essential for the conversion into the sugar, that is cellulose. With a large requirement of this enzyme during production, hydrolysis and ethanol fermentation can be carried out in a single step with involvement of different potent strains. Like we can use the strains such as fungal strain or bacterial strain, and then the enzymatic hydrolysis takes place simultaneously, like microbial inoculation and enzymatic hydrolysis takes place altogether simultaneously to save our time. Next is the glucose is further converted to bioethanol using a potent microorganism, which I have already speak. Then once a potent microorganism, for my work, I particularly choose Aspergillus niger, which is inoculated in pre-treated biomass. It enhances the production of cellulose enzyme, which will ultimately increase the efficiency of biofuel, which will increase the amount of production of sugar, which favors the biofuel production. Then the greater the efficiency of enzymatic hydrolysis, the higher the amount of sugar will be obtained, which ultimately gives us the high amount of biofuel, which is our aim. Then the biological conversion of biomass using cellulose enzyme is the utmost requirement. It is the topmost requirement nowadays because of the increase in rate of fuel, petrol, diesel, and everything. So in reaching an economical model of ethanol production through faced with several challenges like process related complication, heterogeneity of growth, mass, since it is a biological based biofuel, so it require all the things like growth, mode mass, transfer mass, different mechanics, changing rheology of growth during process, microbial growth stages, and so on. And for that, solid state fermentation is reported to have the best result in terms of cellulose enzyme production. But to address a huge amount of submerged culture, process development is the most extensive study at present, that is solid state fermentation. Moving towards my methodology, it will start with the agro-waste selection. Starting the agro-waste selection, like I personally consider sorghum for my further study. Then the biomass need to be separated. It can be separated by different method, that is physical method, chemical method, enzymatic method, biological method. So it can be pre-treated by different method. Chemical method is the most potent way to degrade it because it involves strong acids like H2SO4, sulfuric acid, and hydrochloric acid, and there are so many acids which plays an important role. After that, pH determination should be taken, and because pH favors the pH gives the role like which is acidic or basic. If pH favors the low, then it will be favor the bioethanol production, will ultimately increase the rate of sugar. After that, hydrolysis method. Hydrolysis method can be categorized into two different zone, that is enzymatic method and microbial method. Microbial method, that is also known as biological approach, in which microorganism can be introduced into it. Here is the complete picture. Like generally, bioethanol can be produced from many agricultural sources such as sugar, corn, castor, lignocellulosic biomass, sunflower, maize, wheat, molasses, agricultural waste, sorghum, soybean. But for my work, I preferred sorghum green leaves contain cellulose, hemicellulose, and lignin. The carbohydrate constitute, and there are so many periods in their cell wall because of beta 1 galactosidase bonds linkages are there, which is a kind of obstacle during the production because it cannot allow to degrade the biomass and it cannot allow to convert it into the sugar molecule. Then biomass substrate separation. Initially, it starts with a grinding, like chopped with a small amount. Then it can be converted into the powder form. Then pre-treatment method, that is with different chemicals, but I choose for my work, that is hydrochloric acid and concentrated sulfuric acid. Pre-treatment can be done for breakdown, that is sacrification of complex molecules present in biomass into simple monomers molecule, into simple sugar, because ultimately you have to obtain the sugar. Next is the hydrolysis. Hydrolysis is required to change the conformation of biomass, that is size, structure, and its chemical composition. Just after pre-treatment, hydrolysis should be done. It can be performed by microbial method. There are so many microbial method, like there are so many potent microorganisms, such as bacterial, fungal, and so on, which can increase the rate of production, basically, which can favor the rate of production, amount of production. And enzymatic method is somewhat relevant with that. That in this method, several enzymes directly, like on that, production of enzyme can be taken place. But in enzymatic method, directly inoculation of cellulose or the enzymes which are used for hydrolysis can be inoculated, which is my main motive. So it starts with microbial method. After pre-treatment of biomass, inoculation of microorganism takes place. These are the names of microorganisms, which are also good and suitable microorganisms. But I choose, after going through the several research papers, I choose Aspergillus niger as an appropriate and most potent strain for my work. Aspergillus niger have been extensively studied for the production of cellulase. Basically, it favors the production of cellulase very much. It will produce high level of enzyme which are required for the conversion of cellulose into cellulase. It is a genetically tractable and industrial fungus that proficiently degrades cell wall polysaccharide, which is a major obstacle during production. And it is a graphical representation that when you ferment it, when you incubate it, its production can be monitored during a stationary phase, and after certain period it should be declined, like a death phase. Then cellulose act as a biocatalyst. Basically, once a cellulose can be converted into cellulase, and this conversion should be taken by a microorganism, definitely, which enhance the rate of reaction, ultimately result as an increased in concentration of glucose. And these are the optimizing condition for cellulase production. That is the fermentation method. Definitely, there are 3 fermentation method. Among that, submerged fermentation method is a suitable method for because we are practically in a pilot scale level, so it is the preferable method for that. Carbon source, nitrogen source, pH, temperature, metal ion effect, there are incubation time, aeration, and suitable microbial species, and so on. Estimation of growth kinetics. Once cellulose can be obtained, once a growth can be seen, then this can be monitored by following methods like thread cutting method, filter paper collapsing method, spectrophotometric method, flat bed method, branch and Swan method, CMC method, and everything. But spectrophotometric method is the best method for that quantification. Significance is that it does not produce any kind of harsh chemicals or gases. It does not require very expensive equipment. It does not require large amount of manpower.
Atwari,
Future perspective. Yes.
your time is almost
Hello?
up. Please wind up fast.
Yeah, it's a future process, that is by following the process of production of crude cellulose enzyme in future scenario on diverse industrial process, make it more valuable and demand in production process to be cost effective as well as eco-friendly. It helps to achieve the higher demand of energy in more sustainable way. It may lower the level of atmospheric pollutants such as sulfur dioxide that contribute to the acid rain pollution. That's it. And then references, and thank you. Yeah, that's it. I'm done with my presentation.
Yeah. Really, it's a nice presentation. Are you hearing me?
हेलो
आवाज आ रही है आपको?
Yes, sir.
बहुत स्लो आ रही है, आप बोलिए.
Yes. Now it is audible?
Yes, sir.
Yes, sir. Yes, sir. It is audible.
See, we have seen different kinds of sources of cellulose enzyme. So can you name some of the sources, industrial sources?
Yes. So you're talking about the microorganism or the plant-based sources? Like you're talking about the substrate or the
yes.
organisms, microorganisms.
Yes.
Okay, substrate. So there are in all the green plants, presence of cellulose is there. That is generally I choose sorghum because it contains large amount of cellulose. Also, all the green plants, most of the green plants have cellulose in it.
Okay, right. So in your study, you have used some kind of optimization with the different parameters. So do you use any algorithm for that, for any software, or just how you know optimize those parameters?
Yes, sir. We optimize growth parameter by following the papers, like by following the data of previous research, like what we can do in favor to increase that protocol of previous papers only, and then follow to get a better result of that.
No, can you name any optimization method or algorithm that has been used or studied by you in those papers?
So like Blanket-Bowman method to optimize that methods. Like we obtain the minimum data and then we get that
Okay,
data.
right, right, right. Right. Say one last query is that since you know this is a widely used method on by which you can say hydrolysis you have used. So what are the other approaches that might
Yes.
be possible for this doing the same thing?
Yes, sir, definitely. It has so many other approaches. Like I said in my slides, that it contains the mechanical method, chemical method, hydrolysis method, physicochemical method. So many methods, but biological method is most appropriate because it favors the environment. Like it does not contain any pollutants. First of all, which contain that is almost present in all the fuels which are used at present, right? And that's why biological method is the most appropriate. But the speed of that method is quite slow, and for that, my research is basically to increase that. How would we, what we can do to increase that the production rate?
Yeah. Thank you. Thanks a lot for nice presentation. That's from my side.
Thank you so much, sir. Thank you so much for your questions. Okay,
Thank
thank
you
you.
for the question, sir. Now I would like to request Soumya Srivastav to please start with your presentation and Anika Gupta to please be ready with your presentation.
Okay, ma'am. May I share the slide?
Yes, you can share your slide. Pratibha Tiwari, please stop sharing your screen. Soumya. Soumya, are you having any trouble?
Ma'am, my slide is visible.
No, it is not visible.
Okay, please give me a second, ma'am.
Okay.
Ma'am, now it is visible.
Yes, your screen is visible.
Okay, ma'am, my topic is hello, good afternoon, everyone. My topic is production of xylene enzyme from fungal strain in study of the biofuel. First of all, we know that today is the global pollution is rapidly increasing, and the day by the day consumption of the biofuel or ethanol is also increasing, and the demand is highly rapidly increases. So, by seeing all the demands and all the requirements, the industry will try to like make the eco-friendly approaches so we don't harm our atmosphere or our environment. And for this, I use the enzymatic process for the production of biofuel, in which I use the microbial strain, I use the fungus strain. First of all, xylem is part of beta 1,4-link, which is a class of enzyme, and it is a biological catalyst that used to degrade the linear polysaccharide, that is xylene, into xylose, and also it will help to break down the one of most either component of the plant cell wall, that is hemicellulose, and play a very great role in microorganisms that plant sources for the degradation of plant matter into usable nutrient, and also help in tree bleaching and bioconversion of lignocellulosic biomass substrate. Microbial enzymes. Enzymes are protein molecules and also they are biological catalysts. Microorganisms are the primary source of enzyme because they are cultured in large quantities in short span of time. The sources for the xylanase enzyme protections are by fungi, by bacteria, yeast, marine algae, protozoa, crustacean seed. But by last few recent research, some of best microbial sources of the xylanose production were listed. These are Fusarium, Micrococcus, Bacillus, Aspergillus flavus, and A. niger. In this process, the microbes I choose, it is the flow chart for the microbial organism. Parameter which affect the production rate, it is fermentation method, carbon and nitrogen source, pH, temperature, ionic effect, incubation time, and radiation. Application of the xylanase enzymes are like it is used in textile industry, it will use in biorefinery for use pulp and paper industries, food and feed industries, chemical and pharmaceuticals. Today's in a worldwide, the application of ethanol is using in fuel industry 68%. At industrial level, it uses is 11%. I'm sorry, 21%, and in beverages level, it uses is 11%. The methodology I obtained for this, first of all, I choose agro-based selection, in which I chooses the green varieties. Then I do the biomass substrate separation. Afterward, there's a pre-treatment method, which I apply physical and chemical pre-treatment method by applying milling and microwaving, ultrasonication process, and in chemical treatment method, I use alkaline acid. Afterwards, I use microorganism and do the hydrolytic approach, then fermentation, physical, after there's a physical analysis, and then distillation process will be. The conclusion of this work is basically to try to attempt a research to identify a number of solutions that are friendly and safe energy sources, along with most effective means of avoiding all the negative impact of biofuel, providing the biofuel from the agriculture residue. As we know, agriculture waste is in very high amount and also in trending in today's recent research. It's very effective and environment friendly. So the degradation done mostly by using xylanase enzyme for better production. We work on the production of biofuel by using the lignocellulosic biomass. So we will basically try to focus on second generation by studying all the recent papers and review articles. The future prospects of this through using crude xylanase enzyme in future scenario and diverse industrial process make it more valuable and demand in production process to be cost effective and as well as eco-friendly. Out of different agro-based biomass, lignocellulosic biomass and wet food xylem amended the common it effectively includes the highest and cost effective food xylophoretic enzyme. So thank you everyone, and yeah, I'm done with this, it's done now.
Hello. Yes.
Yes,
Soumya?
sir.
Yes. Very it's
Yes,
a
sir.
nice presentation, right?
Thank you, sir.
Yes, you know that in the case of biofuel or this biodiesel, have you heard about the term biodiesel?
Sorry, sir.
Have you heard the term biodiesel?
Yes, sir.
So can you elaborate different methods for biodiesel production?
So there are various process for the biodiesel production. Like we can in large generation we use as like the substrate our food crops for the production process. In second generation we using the microbial approaches or using agricultural residues. And after also there is a fourth generation and third and fourth generation the fungal or algae method according to the recent research papers found suitable for the like future generations process for the production of biofuel.
Okay, so what are the different examples of microbial enzymes?
microbial enzymes are, it will be like I choose Fusarium sporotrichoids. It may be like by using fungal strain, by using yeast strain, or by using Saccharomyces like Saccharomyces cerevisiae, Aspergillus niger, and many of the other bacteria also will used in for the production in biofuel.
So, what are the different application areas and what are the advantage of using biofuels? What is your comment about this?
Different areas for the using biofuel.
Yeah.
So it mainly it used as a mainly in our domestic level as a cooking, or also in our daily transportation services, and due to like using fuel we are using in our past days, from past days, that will harmful for like harm for our environmental environment as well as our atmosphere. So yeah, today's recent research and scientists and researchers were trying to like find the better and suitable approaches so they don't affect our environment, don't harm our any atmosphere or our like ozone globe layers.
So, have you heard about Jetropa?
Sorry, sir.
Jetropa सुना है आपने Jetropa?
No, sir. I
Okay,
didn't heard about it.
okay. Thank you. That's from my side. Thanks for presentation.
Thank you so much, sir. Thank you for this question.
Thank you for the question, sir. Now I would like to request Anika Gupta to please start with your presentation and Priyanka Thareja to please get ready with your presentation. Anika? Anika Gupta, are you there in the meeting? Okay. Priyanka Thareja, please start with your presentation. Priyanka?
Yes, ma'am, I'm starting.
Okay.
Is my screen visible like shared?
No, your screen is not visible yet. Priyanka? Priyanka, please share your screen.
Is it visible now?
Yes, your screen is visible. You may start.
Good evening, sir. My topic is BRCA gene mutation impact on breast cancer in India. We all know that breast cancer is a major health issue globally and most common cancer in women and even most common cause of death. And it's multifactorial. It can be through environment, lifestyle, hormonal, and genetic factors. And one of the genetic factors is BRCA1 and BRCA2 gene predeposits. They are really it predisposes as an individual to developing breast cancer, and these mutations can be inherited or can be due to can be somatic due to environmental and genetic factors. So what actually is BRCA1 and BRCA2 mutations? So they were discovered in 1994, and they play a key role in our DNA damage response. But when there's a mutation in the genes, they can really be harmful to our body and can cause breast cancer, including other cancers too. Uh, so this is the stat that in general population the breast cancer can be 12%, and in India it's 5 and 8%. But if there's a mutation, then and for BRCA1 mutation it's 50 to 80%, and for BRCA2 mutations it's 40 to 70%. And ovarian, it causes other cancers as well. It increases the risk for ovarian, male breast, and prostate cancer.
rate in pancreatic cancer by a huge ratio. So, epidemiology of breast cancer in India. In Asia, breast cancer incidence peaks among women in their 40s, whereas in United States and Europe, it peaks in their 60s. In India, premenopausal patients constitute about 50% of all patients. It is expected that in the coming decades, these our countries will be the majority of the new breast cancer patients diagnosed globally. Over 1 million new breast cancer patients are estimated to be diagnosed annually in India. A study conducted by the Harvard School of Public Health revealed that 1.35 million cases of breast cancer would be diagnosed worldwide in 2009, accounting 10.5% of new ones, and it will increase by 26% by 2020. So, BRCA gene testing. People who inherited these mutations are at increased risk of developing breast cancer and ovarian cancer compared to the general population. The BRCA gene test is offered to those who are likely to have inherited mutation based on personal or family history of breast cancer or ovarian cancer. The BRCA gene test isn't routinely done in people at average risk of breast cancer and ovarian cancer. The results of genetic testing aren't always clear. A positive result means you carry a gene mutation that increases your risk of cancer and doesn't necessarily mean that you will surely have breast cancer in the coming years. A negative means that you don't have the mutation, or you might have a mutation that doctors haven't discovered yet. Why the test is done? So the BRCA gene test is a blood test that's done to determine if you have the changes, mutations in your DNA that increase the risk of breast cancer. Mutations in either breast cancer with either one or BRCA1 or BRCA2, it increases the risk of breast cancer, ovarian cancer, male breast cancer, prostate cancer, and melanoma. So our conclusion is the prevalence of pathogenic mutations in breast cancer patients is higher in Indian patients compared to with most other population. At the same time, there's urgent need that we improve post-test counseling and extend it to unaffected providers in order to improve the uptake of prophylactic interventions and cascade testing. Thank you, sir. Uh,
Priyanka,
yes,
are you done with your presentation?
yes, ma'am.
Okay. So please ask the questions if you have any. Sir,
Yes, Samiksha. Actually,
sir,
Tarun sir has leave the meeting. Yeah. Actually, Tarun sir has leave the meeting, and Tushar sir, sorry, and he is soon going to join with us. And so just wait for it.
okay, sir. Priyanka, please wait for the questions.
Okay.
Tushar sir, Priyanka has done with her presentation. So if you have any queries, then.
Yes. Hello, am I audible?
Yes.
Yes, sir, you are audible.
Yes. 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 See, in the breast cancer, it's a very, widely spreading disease, So
Yes.
the very first thing that what are the, possible ways by which we can stop this disease in the early stage? Is there any, foolproof method, or we can do that, or after the age of 50 or some study that you have shown, the chances are getting more. But is there any early, preventive measures that can be taken to stop it?
So if it is genetic and our parents had it or my mom had it, so I can get tested regularly for the mutations, and eventually if it is there, one of the possible way to eradicate can be mastectomy. Most of the people who have mutations in their body, they prefer mastectomy. Checking for lumps regularly, checking for lumps regularly in the body, and lifestyle changes. I mean, one should lead a healthy lifestyle. Be it cancer or any disease, one of the major reasons of increasing cancer is bad lifestyle of people, and then it shows in the later stages in the 50s and 60s.
And one thing more, that in today's time, especially the youth are living with the stress. So, in your opinion, whether stress might also cause this disease?
Yes, sir, stress can surely cause this disease. Stress has been shown to increase the possibility of many diseases, and cancer is one of them, and breast cancer is no other.
Okay. Yes. Thank you. That's from my side. Thanks a lot for your nice presentation.
Thank you, sir. Mm. Thank you, ma'am. I'm done with my presentation. You may proceed ahead.
We have one more participant. She will be joining soon as she is having some network issue.
Tushar sir, sorry for the delay. Actually, one more participant is there. She is having some
No
network
problem,
issue.
ma'am. No
That's
problem.
why.
I'm available.
Yes.
No problem.
Okay, thank you, sir. Thank you, sir.
Sir.
Yes, Samiksha? Anika, you can share your presentation. Anika, am I audible to you? Anika?
नमस्कार।
Hello. Ma'am, I'm audible.
Yes, you are audible. Please be a bit louder.
Okay, so today, good evening, everyone. Our topic is cellulose enzyme production from fungal cells for biofuel production. As we all know that we need renewable sources for the ethanol production from the fossil fuels. In the today present world, there are 205 billion liter bioethanol per year was produced from the rice straw, which is total of the 5% of the total consumption. Rice straw prominently contains cellulose between 32 to 47%, hemicellulose from 19 to 27, and lignin to 5 to 24, and ashes 18%. The pre-treatment is necessary of the rice straw to obtain the ultimately high degree of fermentable sugar. Here are some objectives that we have covered. The first one is pre-treatment of rice straw by alkali solution. Second is to isolate trichoderma from soil sample to prepare PDA media culture for fungal growth, and to do substrate chemical hydrolysis and DNS test for the black liquor to determine the reducing sugar, mainly the concentration of sugar. So here the material and method. The first one is the pre-treatment of rice straw that we have done from the alkali method. And in this we have used NaOH, which is dissolved in 100 ml, and then we heat it at the 55 degree centigrade incubator, and then we put it in an oven for the 24 hour at 60 degree temperature. Then we separated the black liquor from it and extract the rice straw for the further analysis. This is the separate step that we have done. After that, we have done the isolation of trichoderma from soil sample. It is done by the serial dilution of soil sample that we have taken from the ground of our college itself. And then we Then we have prepared the PDA media culture for the fungal growth. As we know that for any fungus or any microbes to grow, that we need a media that is probably most favorable is PDA. And for strengthening the growth of the microbes, apart from fungus, we have add antibiotic that is ampicillin 0.3 gram in 1 liter of media. Here is a result of that trichoderma species that we have obtained in the first culture. In the second culture that we had made for the further growth, this PDA slant. We have used a commercial PDA powder to make that plant and use the general protocol for that. And after that, we have done the chemical hydrolysis for the same, for the alkaline pre-treated rice straw. जो हमने पहले sterilized किए थे in 125 ml flask, then we have pre-treated at the first is adjusted to glacial acetic acid, then it is taken to sodium citrate buffer and then added to sterilized water to final buffer concentration of 0.05 molar. Then we have the tetracycline and chromophenicol to prevent microbial growth. Then we have collected the sample and restored in the oven for some dry heating bath time at 105 degrees centigrade for 50 minutes to inactivate enzyme and centrifuge at 5,000 gram for 5 minutes. The result was the presence of alkali during pretreatment of rice straw at 55 degree to 95, increase in glucose conversion ratio for glucose yield from the raw biomass upon 72 hours of enzymatic hydrolysis when compared to the wash only treatment, and the highest ratio in yield of washed biomass from CaOH to pretreatment was 56% weight by weight of the solid fraction of pretreated biomass glucose contained in 183 mg per gram initial glucose contained in the raw material respectively. Then we have done the DNS test of the black liquor to determine the concentration of glucose in the sample. The procedure was then as given in the standard protocol of the DNS test. The result that we have obtained is this here in the graph. Here we get some concentration of glucose that is about 0.925. So we have concluded from that if there is a presence of glucose from the rice straw we have treated, it means that the enzyme was present, that is cellulase enzyme, which is produced when the fungus eat the media to produce cellulose, that is sugar. Then conclusion, but there are several important aspects that we should consider for the development of any process in solid fermentation. Here we optimize the process by parameter and separation of the product in order to achieve high enzyme yield efforts and need to develop a suitable medium and to work out a favorable environment condition for proper growth and maximum secretion of enzyme. Thank you.
Yeah, thank you, Anika, for a nice presentation. Right. See, this biofuel, if you remember in the early, time, it has big a buzzword, but now it has not been used commercially, So what may be the reasons that this fuel is not so popular, by which it is having so much qualities, So how can you comment on that?
So because of the non-availability of the enzyme or the transportation cost, somehow it is more costly for preparing biofuel at the industrial level. But as we have done from the fungus strain that we can obtain from any soil, nearby soil, that it can be very cheap and time consuming. So in the future scope, we can use this process.
Okay, have you heard about Zetropa biofuel?
What?
Zetropa.
No, sir.
Zetropa. It was early biofuel. If you heard about, सुना है आपने कभी इसके बारे में?
Sir, actually, I don't remember. Sorry.
Okay, so can you name different uses of biofuels in different sectors? Different industrial sectors, how biofuels can be used?
At industrial
Any
level.
idea? Yeah, in industrial. Like one I am giving like in transport, another might be.
Sir, it can be used in like for transport, as you say, and secondly for the alcohol production. It is the same. Before the biofuel, we have obtained the bioethanol that can be used for the alcohol production
Yes.
also.
No. In the industries, like you can use it in transportation, aviation, lubrication, oil cleanup operations. There are so many, uses or application areas of biofuels. Okay, thanks a lot. Thanks for your nice presentation. All the best for your future. Thank you.
Thank you, sir.
So thank you, participants, for giving your oral and poster presentations to us. And thank you so much, Tushar sir, for giving your quality time with us. And it's been an immense pleasure for us that you are here and serve as
Same here, ma'am.
as a. So, sir,
And once again, I
I
want to
request all.
thank you all that giving me this opportunity to be a part of this wonderful conference. Thanks all participants. Thanks the organizers. It's my all best wishes to you. Thank you. Thanks you all.
Thank you, sir. And participants, you are requested to fill the feedback form that has been shared in the comment box. So it's a request for all the participants to fill that feedback form. And now you can leave the meeting and you can join back at 4:20 p.m. for the valedictory session. Tushar sir, you also, if you are having, if there is no any time issues for you, then you can also join at 4:20 p.m. for the valedictory session.
Definitely. Thank you.
Okay. Thank you so much, sir. Thank you, participants. You can now leave the meeting.